to know protein-protein interaction. This is Yeast-2 hybrid assay. We performed Yeast-2 hybrid assay to show protein-protein interaction. So this method is based on, as you all are knowing, that transcription factor, eukaryotic transcription factor have two domains. One domain is DNA binding domain, and second domain is activation domain. So transcription factor consists of two domains, two independent domains, but proteins are same. So scientists, what they did, you can see, if that transcription factor becomes active, if both these domains comes together, then the promoter corresponding to that transcription factor will start transcribing the gene cloned under that promoter. In this way, means what? Means if transcription factor half domain here, you can see. So scientists have designed a vector, 2 micron based vector. This is episomal Yeast expression vector. I have already taught you. 2 micron. So this is Yeast expression vector. So in one of the vector, scientists have cloned activation domain coding gene, transcription factor activation coding gene, activation domain coding gene. And in second vector, they clone DNA binding domain, gene corresponding to DNA binding domain of transcription factor. Means half of the gene cloned in this vector, half of the gene cloned in this vector. Now we are not here, our purpose is not to clone that transcription factor. Purpose is to find out our proteins interaction. So we clone, here there is MCS. We clone one possible interacting partner if we have some clues through other experiment that two proteins are interacting. Then we clone one protein, one gene in this vector and second gene in this vector, provided we must have knowledge about. Then this is this protein-protein interaction technique to find out two proteins. There are also high throughput methods. Means we can find out interacting partner from hundreds of possible putative interacting partner. We also use, but here we use this Yeast-2 hybrid assay to find out our known two proteins. So we clone this one gene here, CAGX suppose here and CAGT here. Now this vector will produce our protein conjugated with binding domain of that transcription factor. This vector will produce CAGX with conjugating with this activation domain of transcription factor. Like affinity tag, here transcription factors activation domain will be conjugated with our one protein. Here DNA binding domain will be conjugated with our second protein. So now if both protein will come together, then transcription factor become functional. Otherwise transcription factor have this binding domain and activation domain, so it will not come together, so it will not be functional. So how can we check functionality of transcription factor? So if this transcription factor is active, then it will bind and start the gene expression. Which gene? So we design a yeast strain where there are some reporter gene cloned under the control of this transcription factor, which are cloned here. So in yeast strain, there are some reporter gene. Reporter genes means what? Their expression we can easily observe. We can easily check their expression. If it will express, we can check its expression. That is reporter gene. It will report. So genes, reporter genes are cloned in the yeast under the control of this transcription factor, which are absent in that yeast. When we add this two vector in that yeast, if it will express, then our target protein, bait protein, and this prey protein, if they will interact, so transcription factor will also come together. Means their binding domain and activation domain will come together and transcription factor becomes functional. Now, since transcription factor becomes functional, it will bind to their promoter region and then start transcription of promoter gene already present in yeast strain. Now, if it will produce that reporter gene, means we can find out their presence or absence. If it is expressing, reporter gene is expressing, it will produce protein. So in this way, if it is producing protein, that means the transcription factor become functional. Why this become functional? Because our bait and prey protein comes together. Fine. So this is to hybridase. So we co-expressed these two vector, having one have one protein, target protein, and second have second target protein. One have tag X, one have tag T. And this vector have already activation domain coding gene. This vector have already DNA binding domain coding gene. So if both here we co-transform, so there are selection marker. What is the selection marker? Here you can see where is selection marker. So here leucine is selection marker, here tryptophan is selection marker. So in absence of this vector, yeast strain will not grow without supplement of leucine and without supplement of tryptophan. When we add these two vector in yeast, that yeast strain can grow without supplement of leucine and tryptophan, because leucine biosynthesis will start by this enzyme coded by this gene, and tryptophan biosynthesis can take place due to presence of this gene, TRP1 gene. So we call it again, I have also discussed during yeast artificial chromosome. So this is selection marker. So this is auxotrophic selection marker. When both these vector will enter into the yeast, that yeast strain can grow without supplement of leucine and tryptophan. So you can see here, leucine minus tryptophan minus media we used. That means the transformation is successful. Then our purpose is not only to transform, our purpose is to check whether both are interacting or not. So then there are reporter gene. Reporter gene is here, histidine production. Enzymes which can continue histidine production. So if both the proteins interact, then that transcription factor becomes functional, and then histidine production will start in the cell inside. So if both are interacting, then only we can grow this cell, yeast cell, in absence of histidine. Since this is growing in absence of histidine, that means our transcription factor is functional. Why? Because if that transcription factor become functional, then only histidine reporter gene will express, and histidine reporter gene will express means that yeast strain doesn't require histidine supplement, means in absence of histidine it is growing. Why transcription? How transcription factor become functional? Because they come together. How they come together? Because our both this CAGX and CAG-T protein coming together, so that binding domain and then activation domain of transcription factor become functional. In this way, indirectly, we observe or we determine protein-protein interaction. Here, our target is CAGX and CAGD. You can see negative control. We use this empty vector. We have not cloned any gene, but activation domain we have cloned. In negative 2, this activation domain is empty, and here we clone CAGD. So both are negative. This is non-interacting partner, so we took this as a positive clone. And T1, T2, T3 is 3 colonies from double dropout plate. Double dropout means we have to supplement this leucine and tryptophan, then only it can grow. So we used PGAD CAGX and PGPD CAGD for test. Means in one vector we cloned X and second vector we cloned T. So this CAGT protein will be conjugated with DNA binding domain of transcription factor. CAGX protein will be conjugated with activation domain of transcription factor. If both this CAGX and CAGT come together, then transcription factor becomes functional. And if it becomes functional, then reporter gene will express, which we can observe. Sometimes reporter genes are color producing reporter genes. So we can easily check this to hybridase. And there are other methods also, centrifugation, phase display, there are freight, there are several, but we have all these techniques which I have performed. So you can ask question even if you have performed this or you wish to perform in future, then you can ask this question. But these techniques we frequently use to find out protein-protein interaction. Suppose we now conclude